to continue our discussion, we go now to Erysipelotrix insidiosa and Erysipelotrix luciopatiae. So this is, they cause erysipeloid or red skin. Okay, it has three distinct clinical entities. Erysipeloid localized cutaneous infection usually found in the fingers and hands. So generalized cutaneous infections of the usually found at the cutaneous layer of the skin. Septicemia, microorganisms, so gram-positive rods, non-motile, non-spore forming, and facultative anaerobic. So specimen and media, usually skin biopsy from advancing borders of the lesion. Blood specimen if septicemia is suspected. So we have a culture media of 1% glucose broth and blood agar. Or nutrient broth. So this one is a solution medium. Make gram staining for the organisms from a clinical specimen and growth on the blood agar. Usually ito yung medium that are medium where the bacteria are obtained for gram staining. So biochemical tests used in identification, so test tube brush appearance in gelatin stubs. Okay, and there is hydrogen sulfide productions, acidification of slant and bath in the triple sugar iron agar with no gas production. And then to differentiate organisms from Listeria Monocytogenes, perform the catalase test. So Listeria Monocytogenes is catalase positive, while the luciopathy is catalase negative. So treatment, penicillin is a drug of choice for treatment of erysipeloid or the Erysipelotrix luciopathy and insidiosa. Okay, next. So this is another one is Listeria Monocytogenes. Usually these are found in the meat or lean meat or raw meat. Okay, this is Dysteriosis or Listerosis. It has 4 forms: non-specific flu-like illness during pregnancy and puerperal sepsis. Okay, neonatal sepsis or later meningitis that will affect also the babies inside the mother's womb. Sepsis and meningitis are immunocompromised patients. So usually when you say immunocompromised patients, so the immune system of the patient is already deteriorating or considered to be weak. So food poisoning, microorganisms, gram-positive rods, shorter and thinner, non-spore forming, facultative anaerobic, and beta-hemolytic on blood sugar. So biochemical characteristics, so motile, so umbrella type growth in semi-solid media. So may tumbling motility as seen in hanging drop technique. So usually the bacteria is motile, catalase positive. Specimen is blood, cerebrospinal fluid, and genital tract secretions because it causes meningitis. So pwede yung magkuha ng sample dun sa meninges of the brain. So media is brain human infusion agar plus 5% shift blood. Okay, and we have also the brain human infusion broth or the solution broth or medium. So selective Listeria agar slant SLA or selective Listeria agar A. Brownish to black discoloration is usually around the colonies of the Listeria Monocytogenes. Brownish to black discoloration that is positive for the Listeria Monocytogenes. If you try to see in the physical colony found in the agar. So identification, so demonstration of gram-positive bacilli from stain, smears of CSF and other fluids, food samples. So ang culture and isolation ng blood agar or selective Listeria or SLA, biochemical test, catalase test. Okay, we have also motility tests, serological agglutinations with specific sera. So the treatment is penicillin plus aminoglycosides, tetracycline used as second line of defense or second line treatment patient is sensitive to penicillin aminoglycoside. So, kung sensitive siya, so pwede yung tetracycline. Okay. Yan, pinpoint, small, transparent. Blood agar plate, examine transmitted light. The colonies are surrounded with narrow hemolytic zone. So, there is hemolysis of the blood. Okay. This zone is more easily if seen culture is grown in a thin blood agar plate. On Listeria Monocytogenes, this is the brown-green colored or brownish to black discoloration.